Thank you very much for the kind introduction and the proper pronunciation of my name. I'm not used to that anymore living in the US. um, and thank you, yeah, in general, the organizers for inviting me. So my lab does machine learning for and AI for biomedicine. We develop machine learning algorithms for all kinds of different biomedical data. And I would say we've had and still have an emphasis on genomic and single cell data, and that's mostly what I'm going to talk about today. And specifically, I'm going to talk about learning the language of biology. That's kind of the theme for my talk today. And I would like to argue that biology has a language, in fact, perhaps multiple different languages. And by thinking of it as language, it allows us to use all kinds of new and interesting and exciting tools to model biology and understand it. So to give you a little bit of an example what I mean by language, well, if you think about the immune system, there's highly complex cell communication happening. That's already a language. In fact, the communication in the immune system often happens through small molecules like cytokines and chemokines. And you can think of those as the words. And often these signals don't occur by themselves, but they occur in combination, in different combinations with each other. And you can think of that as sentences. So there is some kind of language that these cells are using to communicate with each other. And understanding this language gives us insight into the biology, but maybe more interestingly, it potentially would give us control over the system if we were able to actually speak this language and we can talk to biology. So for the first project that I want to talk about today is an algorithm that we developed and it was recently published at Nature Methods. It's called CinemaOT. And it's a causal inference framework for characterizing perturbations in single cell data. And we use it to learn some sort of immune language using causal inference. So we have a problem in biology, and that is if we perturb the system, if you modify a system, you can only measure it once. In an ideal world, we would take a system, we measure it, then we perturb it in some way. Let's say you give it a drug, you knock out a gene, or you do whatever with it, and then you would measure it again, because then you know it's the same system. The problem is, generally, measurement is destructive. For example, in single-cell RNA sequencing, when you measure a cell, you have to destroy it. There are some technologies out there that are not destructive, but I would say the most interesting ones, the ones where you have a high-dimensional view, measurement of the system, you have to destroy the cells. So in this project, we set out to create what we call a computational time machine. Basically a way to, through inference, measure a system, and in the process of destroying it, turn back time, measure it now in a different condition, and combine that information. So this is not possible physically, but computationally it is. And the specific solution that we use is called the potential outcome framework, or that's the mathematical causal inference framework that we're using. The idea is that we infer counterfactual cell pairs. A counterfactual is like asking what would have happened if. It's like the time machine that I was just talking about. So we use ideas from causal inference to infer these counterfactual cell pairs. And specifically, we do that by matching cells on confounder signals. So if we think about the data, we can split the data, the signals in the data, into confounder signals and outcomes. So the outcomes are all the signals, for example, gene expression changes, that are correlated with the perturbation, with the experiment. Everything else, all the other variability, we call confounders. If we remove the outcome signals for a minute, we can match on the confounders, and in theory, in confounder space, the two distributions are identical, and then we can use optimal transport to match these two conditions and obtain counterfactual cell pairs. And the way we do the matching is using optimal transport, which is a way to morph one distribution into another distribution. So this is a graphical visual representation of our algorithm. So first, we take our gene expression matrix. We split it using independent component analysis into our confounders and outcome signals. We set the outcome signals aside for a minute. We use optimal transport to match cells between the two conditions on the confounder signal. So in theory, the two distributions are identical and we can do proper matching. This gives us our counterfactual cell pairs. And now we can bring back the outcome signals and ask at the individual cell level, how did the cell respond to the perturbation? So the outcome is the ability that we can ask this question: how does an individual cell, so it's truly single cell, respond to a stimulation? Whereas previously, even though it was single cell data, you can only ask how does the distribution shift, but now you can track an individual cell in time. So for a minute, I want to go back to the idea of language and talk about what is the fundamental building block of language. And I would like to argue that it's synergy or interactions between words. The meaning of a word depends on the context. That is obvious. So there's all kinds of interactions between words. And you can kind of think of the simplest building block of language as sort of synergy between word pairs. And this is something that we wanted to explore in this project. Namely, we wanted to ask, in this cytokine language, in the language of biology, are there synergistic interactions? Are there interactions between the words? Now, our framework, our Cinema-OT causal inference framework, actually allows for asking or answering this question. Namely, we can measure synergy effects. We came up with a synergy score. The way we do that is we can measure perturbation A, we can then measure perturbation B, and finally, we measure A and B together. And then we can ask, when you measure A, B, and B combined, is that different by simply adding up A and B separately? In other words, is there a nonlinear or synergistic effect? Is there an interaction between A and B? And we can quantify this in a score. My amazing collaborator, Jeff, next performed an experiment where he took immune cells and stimulated them with various cytokines as well as cytokine combinations. Here you can see some of the cytokines that we perturb the cells with, as well as their combinations. We performed our causal inference analysis, and the first interesting thing that we found is that even though there's many different cells and cell types, there's only 4 groups of responses. In fact, if you look at a single perturbation, for example, in this case, interferon beta, even though there's many different cell types, there are only 2 classes of response. So there's many different cell types that are different, but they respond in similar ways. And finally, we did our synergy analysis, and we actually discovered that there is a combinatorial logic. So we looked at the cytokine combinations and asked, is the effect when you combine them different from when you perturb the cells individually? And in some cases, it was a vastly different effect. For example, in particular in monocytes, as you can see on the right, there is a huge synergy effect, and we can actually quantify and we quantified this effect, and we can see that when you combine, for example, interferon beta and IL-6, that there's a huge response that does not exist when you treat the cells individually. Okay, so this framework allows us to kind of quantify, investigate the most simple building block of language, namely synergy, interaction between words, if you will. In the next project, we wanted to take this to the next level and really try to learn what we call the language of biology. And this project is called cell-to-sentence. So how do we get language from single-cell biology? Well, we can think of genes as words, we can think of cells as sentences, and we can think of tissues as paragraphs or documents. That's how we thought about it. Now, understanding the complexity of biology is really hard. Biology is diverse, biology is complex. There's many different data modalities one can measure, including various metadata. In addition, biology happens at many different scales. There's molecules, there's cells, there's tissues, etc. How do you make sense? How do you model this vast complexity of biology? Well, to do this, we present cell-to-sentence. This is our solution to this problem. Cell-to-sentence allows transformation of single-cell data and other data into a natural language format, such as you can use large language models, existing pre-trained large language models, to model single-cell data. So why would we want to do this? Well, there's many advantages. First of all, LLMs have already some biological knowledge because they have seen data, text on biology. In addition, they have natural language capabilities for understanding and following prompts for diverse tasks. And they're really good at synthesizing multimodal information and ingesting it and reasoning over diverse data. And this was our reasoning for using LLMs to understand biology. So the idea is simple. You take your single cell data, you convert it into a textual format, in our case by rank ordering the gene names by decreasing expression. You run them through LLMs, do various tasks, you can even generate cells, and you can transform them back into the data. So recently, there have been several foundation models, single-cell foundation models that have been presented and published. The advantage of our approach is that we can make use of autoregressive pre-training, as well as do conditional generation, and we have multimodal flexibility. So this is kind of the vision for our foundation model based on cell-to-synthesis. So we take lots and lots of single-cell data, including non-single-cell data, various biological annotations in the form of text. We all feed into one model, and then we can do many different downstream tasks, such as generating cells, predicting labels, generating text from cells, predicting perturbations, etc. So the foundation of our model is a cell sentence, which is the rank ordering of gene names according to a decreasing expression. So you just take the gene names and order them by decreasing expression. And it turns out you actually preserve the vast majority of information. So by just simply taking the rank of the expression, you lose very little information. So you can see this in this plot. There's an R² of 0.85. So you lose very little information by transforming to a rank and then transforming back to expression. So we can even take generated cell sentences and transform them back to expression with very little information loss. This is kind of an overview of the framework. So you can take gene expression, turn them into your cell sentences. You can add all kinds of textual annotation. Then you do LLM fine-tuning. Then you do various downstream tests, such as generating cells from text or generating text from cells or other tasks. And then finally, if you have generated cells, you can transform them back into expression and just work with your regular gene expression matrix as you're used to. So now that we can train and fine-tune LLMs on genomic data, now we can define single-cell tasks in terms of natural language. It's basically becoming a prompt. So this is some examples of how you would use the system. For example, if you want to do conditional cell generation, you give a prompt, generate genes for this and this cell, and it generates the cell sentence. Or you can ask it to predict labels or do natural language interpretation. So one of the advantages of our approach is that we can take large pre-trained language models. And you might wonder, well, why is that an advantage? Natural language is very different from biological language. Well, turns out that it's not. We find actually very strong result that using a natural language pre-trained model hugely boosts performance in the cell sentences. So even though the languages are vastly different, it helps a lot that the model has seen natural language and then learns this biological language as opposed to learning biology from scratch. So that's already one advantage. We were also able to show that we can generate meaningful cells. So for example, we can generate cells conditioned on cell type or perturbation. And here's a visualization of generated cells. So first of all, you can see, at least in the UMAP, they look very similar to the original cells. And we can quantify this, and we actually have benchmarked this very extensively against all kinds of other generative single-cell approaches, such as SCGen, SCVI, as well as more recent approaches that are also foundation models, such as SCGPT, and we outperform all these methods. So we can also do perturbation prediction. So in this case, we generate cells based on cytokine stimulation. So this is actually the same data that I presented before. This is an example of the prompt. So you give it a cell type, you say, this is the stimulation, and it generates the cells. And again, we outperform other models. We can also predict complex labels. And interestingly, we can do this not just in single cells. We can create cell sentences from bulk expression data. As long as you have gene names and expression levels, you can feed it to our model. So we're able to run our model, for example, on L1000s, which is a large cell line perturbational data set, as well as GTEx, which is different tissues. Both are bulk RNA-seq. And again, we outperform other methods after extensive benchmarking. So when you think about modeling cells and natural language, well, in natural language, we don't train models just on individual sentences. Large language models, such as GPT, are trained on paragraphs, documents, sometimes even whole books. Because, as I mentioned before, the meaning of a word depends on its context. Well, the same holds for a sentence. The meaning of a sentence depends on its context. So why shouldn't we think about biology in the same way? So what we did is build the first multi-cell foundation model. Instead of modeling individual cells, we put multiple cells from the same context, for example, from the same sample or the same tissue, into the model, such that we can allow the model to learn interactions, not just between genes in a cell, but between different cells. And perhaps unsurprisingly, this hugely boosts model performance. So in this case, we did a tissue classification task, and we compared models with a single cell, one cell, two cells, and three cells and 5 cells. And the more cells you put into the context, obviously the more information there is, the bigger the performance, and there was a huge benefit. We even were able to train a very big model, namely, this is a GEMMA model of 2 billion parameters. Again, this is perhaps not surprising that when you include multiple cells, there's more information, but we were the first to do this. So for the next application of this technology, some of you might not be happy with, because I'm gonna take your jobs away, because we're going to automate the bioinformatician. The vision is to go directly from data to natural language insights completely automatically. So when you think about it, as bioinformaticians, that's what you do, right? That's the work you do every day. Someone gives you data, or maybe you measure it yourself. You run all kinds of analyses, but the end goal is to get your paper, in Cell, Nature, Science. That's the end result. Of course, there's a lot of work in between. We were wondering, can you automate this with language models? So this is kind of the vision. You have your data, you feed it you turn it into cell sentences, you feed it to an LLM, and then you ask it questions. You ask it to interpret the data. Okay, so how do we do this? How do we build a model that can interpret data? Well, we realize there's, in general, 2 sources of annotation, of natural language annotation. So linking genes to natural language. One is papers and abstracts, right? If you have a data set that's published, there's an associated paper, there's the text, that's the interpretation, and there's an abstract also. But actually, the vast amount of biological knowledge, in a sense, in databases, is in terms of gene sets. So what are gene sets? Gene sets are simply natural language annotation, for example, Parkinson's disease, and then a set of genes, for example, 100 genes, right? It means that those genes are associated with Parkinson's disease. And there's, hundreds of thousands of gene sets out there distributed over multiple databases that provide this information. So we realized that is the information that we need. That allows us to link the gene expression information, the genes, to the natural language interpretation. So we set out to train a model where we combine cell sentences, so the expression-ranked gene names, with annotations, both abstracts and gene sets, and we train a model that then learns to associate the two. So it turns out that there are many gene set enrichment algorithms out there. Some of them have been designed for single cells, but actually none of them were really what we were looking for. So we actually had to develop our own single cell gene set enrichment algorithm that uses a graph neural network to map gene expression so genes to gene sets, and then decodes it back into gene sets, sort of an autoencoder fashion, and tries to reconstruct, which allows us to map gene sets to individual cells. And with this data in hand, plus the abstract, we were able to create this large data set and train our LLM on. So what can we do with that? Well, for example, we can, as I mentioned before, generate textual insights from data. But there's other things that we can do that are, I think, quite interesting. For example, we could try to improve gene sets. We can basically ask, okay, what do you think are the genes that are supposed to exist in this set? And maybe we can improve them. But also we could use this model sort of in a generative sense. To traditional ideas, you go from data to gene sets to interpret it. What if you flip it around? What if you give it gene sets and it generates the cells, it generates the biology that's associated with that? So to show you that, so this is very much a work in progress, but to show you that it's already working, we trained a model to go from data to abstract. So we combined data sets and the corresponding abstracts, and we trained a model. And then at Inference, we gave it a completely new data set. So a data set it has never seen before. And we asked the model generate textual insights. And this is what it produced. So at the bottom, you can see the generated abstract. On the top is the original abstract. And I can tell you that they're very similar. And we can quantify this. We quantified semantic similarity between the generated abstracts and the originals. And we're able to benchmark this. Now, it was actually pretty difficult to benchmark this because what do you compare this against, as aside from humans? So actually, we compared it against regular language models that have not seen cell sentences and asked them to interpret the data. If you have ideas how we can better benchmark this, we would love to hear. But the bottom line is we have strong evidence that we can go directly from data through insights using our cell-to-sentence framework. Okay. Now I want to switch gears a bit and talk about flow matching using language models. So I guess not too big of a switch. So in the first talk today, James talked about generative biology. And I think, that generative biology is becoming a pivotal approach in computational biology. So there's two ways you can approach, understanding a system. Either you measure data from the system and you analyze the data, or you build a generative model that can replicate that system. In generative biology, that's the latter. You train a model to to simulate that system, if you will, to generate data from that system. More specifically, the aim is to sample from an intricate, complex target distribution, for example, cells or proteins. And the project that I'm going to present to you today is called Calm Flow, and it merges flow matching with causal language models to address these challenges. So generating biology, creating these models is very difficult. So the goal, again, is to sample from some complex target distribution. For example, you want to generate cells or proteins. However, it turns out that sampling directly is not possible. It's too difficult. So one solution is actually to avoid direct sampling and do it in a different way. And one solution for this is to model a generative process. Specifically, we can model a process that goes from a simple parametric distribution, such as a Gaussian distribution, into your complex target distribution. And once you've modeled this process, you can now sample from your simple distribution and then generate data points in your complex target distribution. And several approaches in this kind of philosophy have been developed. You're probably familiar with diffusion models. So the state-of-the-art image generation models like DALI and stable diffusion, et cetera, are diffusion models. There, the idea is you take images, you add noise to them, and then you learn the reverse process. And at inference, you can sample noise and generate images. There's also normalizing flows, which learn a series of invertible transformations from noise to data. And more recently, flow matching has been presented. And flow matching is actually based on normalizing flows. And I'm going to talk about that next. So in flow matching, it's a generative modeling technique that, again, transforms a simple initial distribution into a complex target distribution. And it's based on continuous normalizing flows. So continuous normalizing flows are normalizing flows implemented with neural ordinary differential equations. But the main idea in flow matching is that you can train these normalizing flows using what is called a simulation-free approach by learning a vector field that represents the flow of your data. At inference, you then use an ODE solver, an ordinary differential equation solver, to integrate that vector field such that you can sample a noise point, you integrate in time, and get to your target data distribution. So what's this vector field? What does it look like? Here you can see a simulation or a visualization of a vector field in time. So the vector field determines at every time point how the data flows. So the vector field is what you learn, and at inference, you integrate. So the black points here, the black axes are actually data points that you then simulate to get to your target data during the sampling process. So there's some advantages in flow matching. It's quite efficient. For example, you have faster training and sampling compared to, for example, diffusion models. It's quite flexible. You can use different types of probability paths. You can even do optimal transport paths. And it can be stable, especially compared to score matching methods. And as such, people have used flow matching in generative biology, for example, to generate cellular dynamics or to do protein folding. And you can see some example papers here. However, this ODE-based flow matching has some drawbacks. It can have high computational complexity. ODE solvers can require significant computational resources. And also, when you work in high-dimensional data, the complexity grows exponentially. Also, ODEs can suffer from numerical instability. They can be very sensitive to initial conditions, and they can lead to inaccurate transformations. And in general, they have limited flexibility. So our solution is to move from ODEs to IEs, or integral equations. So instead of modeling a vector field, we model an integral over time. You can think of it as the history. This allows us to capture the cumulative effect of past states, so the memory, the history, on the current state. There are several advantages over ODEs. It's a more flexible and expressive framework. It handles long-range dependencies, and there's increased numerical stability. And the interesting thing is that we can take any ODE and model it as an integral equation. We kind of make use of that. So just to give you a little bit more background, so in ODEs, ordinary differential equations, we're modeling instantaneous behavior through this vector field. So you focus on local instantaneous dependencies. There's no long-range interactions, there's no memory, there's no interactions like in time. On the other hand, in integral equations, we model an integral over space and time, capturing the entire history of the system, as well as long-range spatial dependencies. So mathematically, ODEs are described by this differential equation where you model the change of the system as a function of time, whereas in integral equations, the system depends on this integral over time, or space also. So the definition of integral equations is there are equations in which an unknown function appears under an integral sign. So you're learning a function that when you integrate it, you get your result. So they have found many applications in physics, engineering, and biology to model systems with memory as well as long-range interactions. Interestingly, more recently, connections have been made between integral equations and transformers and self-attention. So to kind of give you an intuition, if you look at self-attention, for example, here in this sort of language model, self-attention models interactions, in this case between words, but let's say you have visual transformers, it can be between patches or regions in an image or pixels. But basically the bottom line is that self-attention models interactions between all the components in your system, which actually means that it's implementing or it's approximating an integral over your spatial temporal domain. In fact, when you look at causal attention models, for example, if you think about next token prediction, you only integrate over the history, not the future, because you're only predicting the next token. So that token depends on the history. It turns out that that is a specific kind of integral equation called Volterra integral equations, which have the integral up to t. So they only integrate over the history. So this is a connection that we have made that effectively causal language models implement a Volterra integral equation. And because we can take any ordinary differential equation, which is an initial value problem, we can turn it into an integral equation. We set out to implement flow matching with causal language models. And our model is called CalmFlow, which stands for Causal Language Model Flow Matching. So it utilizes pre-trained LLMs that are already smart, that have been pre-trained on text, it's your regular LLMs. They enable, as such, generation from complex natural language problems because they retain their ability to understand natural language, and they're highly robust and highly flexible. Okay, so how does this work? So in conventional flow matching, when you model the vector field with an ODE, instead we model the whole trajectory with an integral equation. So you can see here that we tokenize the space, for example, in time. We feed that to the LLM, so the trajectory, and then the LLM predicts the next state. So specifically how this works is that we can do condition-based generation, so we can sample a noise point in addition to a natural language prompt. We can then use the LLM to process the flow, so generate the time points. And finally, to retain stochasticity, we actually implement a variational autoencoder decoder step. So if you think about large language models, they operate in discrete time and space. But let's focus on space for a moment. So what is the discrete space large language models operate in? Well, it's the vocabulary. You have a finite set of words, and an LLM at every time point, every token, or it predicts, it samples from that space. In our case, we actually don't want it to be discrete, we want it to be continuous, because often in biology, you're dealing with continuous systems, for example, gene expression in cells, or protein conformations, etc. So we implemented the variational autoencoder that it still allows modeling of this continuous next token distribution, except in a continuous manner as opposed to a discrete manner that LLMs natively do. So what does this look like? Well, we take a system. For example, in this case, we want to generate single cells. We generate trajectories by randomly matching cells to points in our noise space. That creates trajectories. We tokenize those trajectories in time and in space, as I'll show you later. And then we train the LLM to predict these sequences. We can do this in single cells. We can also do this in proteins. For example, if we're interested in generating protein structures, do protein folding, if you will, you can model trajectories from random configurations, random structures to folded structures in the same manner. So I mentioned that actually what we do is not just time tokenization, but space-time tokenization. The reason for this is actually initially we mapped every time point, so whether it's a cell or a protein, to one token, and this didn't work well. Then we realized, well, we don't just want to integrate in time, we also want to integrate in space. We want to allow for spatial interactions. So instead what we did is we did space tokenization as well. So if you have a cell, we cut it up into pieces, if you will. We learn those pieces and distribute the cell over multiple tokens. The same for the protein. Well, the protein has a natural sort of ordering, the amino acid sequence, so we just cut it up into pieces, one or several residues per token. And then we have our space tokenization. And then we model the trajectory in time. So we get this matrix, if you will, of tokens, space by time, and then we linearize it, or we flatten that to create a sequence such as you can feed it to an LLM, such that it can do simple next token prediction. So simply how this works, we do our space-time tokenization, we flatten it into a sequence, we feed it to our LLM generates next tokens, it uses a variational autoencoder to do it in a stochastic manner. Then we sample those points and we train it using a reconstruction loss. Is it predicting the correct token? But we also have a Kale divergence loss because it's a variational autoencoder, and you want to ensure that the latent distribution is well behaved, is close to a Gaussian. So what can we do with this? So I want to show you some examples, some of our results. So first we applied our model to simple toy data sets. For example, on the left, you can see it's generating some sort of Gaussian distribution to 2 moons. It works really well. And on the right, we were generating images. And there's 2 interesting things here. First of all, because we are modeling this process in a latent space, namely the VAE latent space, we can actually visualize the latent space here in 2 dimensions in UMAP, and you can actually see how these trajectories go from noise to some target distribution. The other interesting thing is that we were able to demonstrate the abilities of our model to condition these flows on natural language. Because if you remember, these are pre-trained LLMs, such as GPT or Llama or whatever. So in this case, we gave it text representing the digit, in this case, a very simple example, and we're able to generate flows from those digits. So you say, basically you can say, generate an 8, and it will generate an image of 8, generate a 6, etc. So again, this is very simple, but you can imagine that this can be very complex natural language prompts combined with this continuous flow matching generation. Another application is protein backbone generation. The idea here is that you go from random structures to folded proteins. Those are the trajectories. And again, we did space-time tokenization. I explained this before. So each residue or a couple of residues would go to an individual token. So we have a tokenization in space, and then we have a tokenization in time where we're modeling this trajectory that goes from noise at T0 to the folded structure at T1. So this way you can generate a sequence of tokens, feed it to an LLM. The LLM can simply do next token prediction using the VAE. We have a reconstruction loss, a Kell divergence loss, and then when you generate a new protein, you can then generate a sequence, decode that back to whole proteins in time, and you have your result. So does it work? Yes, it works. So here you can see an animation of a generated protein where we go from noise, random configuration, into the target state, the folded state. On the bottom, you can see some more examples. So this is very much a work in progress, but what we're already finding, which is interesting, is that the traditional ODE-based flow matching generates proteins that are very close to the training data. So you can think of it as overfitting, essentially. Our approach, for various reasons, generates much more diversity, and we think that we have much better generalization power. Also, especially I think in low data settings. And the reason for that is that our baseline model is an LLM. It's already smart. And then we're training it, or we're fine-tuning it to generate proteins. Compare this to a model that you train from scratch. It's much more likely to overfit. So there's a lot to explore here. But finally, I want to show you this result, is where we applied this calm flow model, causal language flow matching, to perturbation response prediction in single cells. So we were actually quite surprised how well this works. This model is really good at generating cells from perturbations. So going from noise to target cells. We are able to generate highly biologically plausible cells. So for example, here you can see on the right, in the blue cells is the ground truth data, in red the generated cells. We outperform other methods, including the traditional flow matching, as well as specialized single cell generative approaches. And what's interesting is actually that we found that increasing the number of spatial tokens, so splitting up a cell into multiple tokens as opposed to one, has a huge advantage, as you can see here, 10 spatial tokens versus 1. So to summarize, I presented CinemaOT, which uses counterfactual analysis and causal inference to quantify perturbation effects. And we made the first step towards learning a language where we're understanding or quantifying synergistic effects between different perturbations, specifically cytokines. Then I presented cell-to-sentence, which is our foundational model framework for converting genomic data into textual format such that we can fine-tune large language models and do many downstream tasks. And finally, I presented calm flow, which matches flow matching with large language models that allows generative biology, continuous generating of continuous systems, but retaining the capabilities of large language models, such as generating these continuous flows conditioned on natural language. And with that, I thank you. And I thank my sources of funding, and especially the amazing trainees in the lab that did all this work, and my collaborators. And thank you all for listening. And let me know if you have questions.
Thank you so much, David, for the impressive work. For questions, there's a microphone there, or you can line up for my microphone here if you're close to this area.
Hi, David. Thank you for that talk. I have a question about your perturbation modeling efforts. So single cell technologies tend to be destructive, so you're never going to get data for the same cell for more than one condition. So given that, I do not yet understand well how you validate the results that you get, your post-perturbation results.
That is an excellent question. So you're right, there exists no data where you can measure the same cell twice in an experiment to perfectly validate the system. So Basically, our validation was twofold. One is we did a synthetic data setting where we simulated single-cell data under various conditions where we do have the ground truth. So the advantage is we have ground truth. The issue is it's still a fake system. And then we did real experiments where we didn't have the perfect single-cell validation, but we could validate the findings, the discoveries that we made. So that's kind of how we approached it. Yeah.
Thank you.
I hope that answers your question.
Hi, fantastic talk, by the way. My question was, it seemed like all your models have been trained on human cells. And I was just wondering if that was the case, if you've tried out whether they perform on something like E. coli or yeast, or if you need to train a special model for them.
So yeah, great question. What I presented here is all human, but there's a lot of mouse single-cell data. In fact, I didn't mention it, but one of the downstream applications of our foundation models is cross-species translation. We're currently training models on also mouse data. And actually, hopefully, if training finishes in time, we'll be releasing several new large models that are trained on human and mouse. So I don't think, E. coli, bacteria, or yeast, there's any, significant single-cell data out there. So we're not doing that. We focus on, I guess, where the data is, which is the mammalian systems. Not just mammalian, I guess, but multi-cell systems. Also, we can't build a multi-cell model of yeast because there's no multi-cells. So.
Thanks a lot for the inspiring talk. I have a question regarding the evaluation strategy for cell-to-sentence. So in a conventional model, you would try to evaluate it on a completely new data set, which the model has not seen before.
Mm-hmm.
But over here, we have the challenge that we give the data as evaluators about the system that we already know a lot. So like there's lots of background knowledge in the field that the large language models were able to grasp.
Correct.
So in a sense, this is not fully independent evaluation, because they're kind of building knowledge and then doing some experiment, but the model already knows the underlying background.
Yeah.
So what's your take on that?
Yeah.
How to evaluate?
So this is a general problem in LLMs, is that there's so much redundancy in the data that, often whatever task you give them, and the model might have already seen that. So you can cheat, if you will. So we have the same issue with biology. There's a lot of redundant information. But there's several ways to fix this. One is to ensure that you are holding out information that's not redundant, where there's no redundancy. So kind of you create holes in the data manifold, if you will. So you know for sure that a model has never seen this cell and this disease or tissue, whatever, before, and then you have a proper validation set. Another way is to do downstream tasks where you also know it hasn't, like you do some kind of perturbation prediction, and you know that the model has never seen that data because no one has measured that before. So again, I think it's just a matter of holding out the right data. Yeah, I think that's probably the most reasonable approach.
Thanks a lot.
Maybe one final question from me, David. So you started your talk by motivating, for example, from this immune example where you have a lot of interactions, cells talking to each other. So why didn't you look at graphs, for example, as a way to model interactions between biological entities?
That is a great question. And I would say, in general, we do a lot of graph work, many graph-based projects in the lab. I just haven't presented them today. So it's definitely very much part of our toolkit, our arsenal to ask these questions. And I think, I mean, people have done this, right? Have tried to model cellular interactions, like these papers like Cell Phone DB or whatever, where they model interactions between cells and signals and cytokines and kind of build graphs to model that. I think that's very interesting. In fact, a lot of biological data exists in graphs, like gene ontology. So we've been thinking about how do you enable large language models to reason about that, there's actually recently quite a bit of work to convert graphs into a format that large language model can reason about. So this is very much on top of our mind, and we understand that thinking about these systems in terms of graphs is important and very useful, and we're doing that.
Great. So let's thank David